as you guys have been working on your quizzes, I've been asking you a lot of questions about whether or not something will come into the cell or will leave the cell. But I think it makes sense right now to think about why we even call them cells. So if you go out in the regular world and you say cell to somebody, they often don't think of like a biology cell. They think of a jail cell or a prison cell. And then sometimes the second thing they think about, or the third thing, would be maybe a biology cell. And in fact, the name cell for the thing in biology came from some of the people that originally kind of discovered that they existed. And those people were monks. So they were not in prison. They were actually thinking of a cell as a room where you close yourself away from the rest of the world and you study and you think about God and you do writing and all that stuff. And now we have this, connotation with cell where we don't typically think of that because there aren't a lot of people in the religious life. So we tend to think in the real world, a cell is like a prison or a jail cell. And then in biology, a cell is this thing that we don't always have a good handle on. So I think if we think about why they got named cell to begin with, what was it that reminded these monks of their chambers where they studied, it can start to help us figure out why something would come into or out of the cell and what can and can't go in. So if you think about the purpose of a prison cell, it's to close in a space. It's to keep some things in. It's to keep some things out. So just like with a prison cell, in a biology cell, we have inside and we have outside, and then we have the membrane in between. And the membrane is what's making that separation. It's what's dividing the rest of the world from the contents of the cell, from the processes and the things that are all happening inside the cell. And that's deliberate in the case of a prison cell. It's designed that way. And in the biological cell, it has just occurred that way over time. But the effect is fundamentally the same. Now, when you look at a prison cell, you realize that the surfaces are not all made out of the same thing. So we've got kind of 3 walls over here that are made out of bars, and then the back wall looks like maybe concrete. So if you look at this cell and you start to think, what does this cell effectively keep in? What does it keep out? And then what things are free to pass back and forth? So if you're a person and you get put in this cell, it is a barrier you cannot get through. So for you, you are trapped in there. You can stick your hands through the bars. You can maybe put part of a foot through the bars. You can stick your nose through the bars, but you're not going to get your whole self through those bars unless you modify them in some way, break them down, destroy them. You're stuck in there. Now, the surfaces of that cell are different. The back wall is concrete, so even fewer things can get through the back wall than through the front and the sides. Because the front and the sides, there's a slot where you could put your hands through to have the cuffs put on. You could potentially have food put through there or medication. You can definitely get air through the sides of this cell because the bars are far apart. Light can get through, but if you look at the back wall, the concrete wall, light can't get through that. So if it's light outside this building, you'd never know it inside that cell because the light isn't able to get through there. So we have to think the same thing with a biology cell. So we know that a membrane in cell biology divides the inside of the cell, the world in there, from the world outside. And its role is similar to a prison cell. You want certain things to stay where they are, but then you have to let other things through. And we have to kind of consider what are those things that can get across in a cell membrane. Now, we've talked about the sorts of things that are capable of moving across a cell membrane. And just like with your jail cell, so air can get through, light, which is not a thing, but light can get through. So those kinds of things, you can get water through those bars, food. So all those things, yes, can get through those bars. But people is a big thing. So people, no, cannot get through those bars. So over here with our cell membrane, we've talked a bit about things that can easily go through the membrane itself. So there are things that can just kind of weave their way right through here, and they don't actually see the cell membrane as a barrier. So just like in a jail cell, air doesn't get interrupted by the bars. It just flows right through. So things that can flow right through a membrane, that can just like come through or can leave, so this is a two-way street. These are things like O2, CO2. So what we're looking at here is non-polar things. So the things we've talked about that are non-magnetic, they can come and go as they please. because they don't even register that that cell membrane is there. The situation that we have, though, is that polar things, like water, for instance, they aren't going to just drift across that cell membrane. Because when we look at the situation with polar things, so let's go to a new slide. If we look at polar things, we've got our cell membrane kind of lying through the middle here. We're just going to super abbreviate it. We know it's got its lipid tails in the middle, and then it's got the heads out here, so we're just going to abbreviate them just to remind ourselves that they are there. And then when we look at the situation outside the cell, so we've got all of our water molecules moving around there, but the inside of the cell is also watery. So we've got water inside and outside the cells. And all those water molecules feel that attraction to each other. They're all magnetic. So we are showing their little, pattern of attractions there. So all water molecules are polar, and so they're attracted to all other water molecules, which are also polar. So you've got all these magnetic attractions between polar things. And when you look at what's happening with the cell membrane, the middle of the cell membrane is non-polar. It's non-magnetic. And so that means there's going to be no attraction between the inside of the cell membrane and any of the waters outside or inside. They're going to be busy drawing towards each other, and they're not going to be particularly interested in the inside of that cell membrane. You will have water molecules that are attracted to the heads parts of the cell membrane, so there will be kind of an attraction right there, right there. And so we can kind of think of it as, all right, so water molecules are attracted to other water molecules, so wouldn't this water molecule be attracted to that water molecule? Like, wouldn't that happen? And when you think about this, you kind of want to think of it as like we've got a bunch of magnets out here. So we got magnets on the outside of the cell because those are the water molecules. We've got magnets on the inside of the cell, more water molecules. And then we've got this cell membrane in the middle that's kind of acting like a block of wood. And if you imagine that a bunch of magnets are kind of hanging out here and then a bunch of magnets on the other side of this block of wood, the wood is keeping them so far away from each other that they really can't feel each other. You're going to feel your attraction to your close neighbor here more than you are to this one way over here. So there's a distance issue that's going to prevent those things from feeling each other. And so when we look at what forces are pulling on the water molecules, well, they're going to feel an attraction like to each other, like all up in here, more so than they are to anybody over here. These guys are all going to be attracted to each other as well. So we've got this problem where water on its own isn't going to cross the cell membrane. It's going to be too busy being drawn to other water molecules and other polar things outside the membrane, and then the water molecules inside are going to be too busy drawn to each other down there. And so it's like they're never going to meet because there'd be no reason, nothing's ever going to pull them in here. There's no magnets in there, there's nothing polar in there, so there's nothing to draw them into the cell membrane and get them to cross. So they're going to tend to stay where they are. But the problem that we have is that you guys have been working on these models where, depending on what fluid you put the cell in, cells can either, so if you have a red blood cell, they kind of look like this, almost looks like a smiley face. They can look like that, but if you put them in another kind of solution, they can swell up. That can happen. And then you put them in a different solution and they get all shrivelly and they look sad. So we have to look at what's going on as the cell changes from a pretty happy, stable cell to a big swollen cell, and then possibly even like blows up, like kablooey. So cells can explode that way. So we've seen that happen because you guys watched a video of a red blood cell being placed in various different solutions, and then sometimes swelling up if it was put in a certain solution, sometimes shriveling up, sometimes swelling so much that it exploded. So we have to think about what's going on there. So if we look at our red blood cell, and we figure it's going to puff up, then we added something, right? It's got more stuff, added stuff. If we take the red blood cell and we look at it when it shrivels up into this sad looking little thing, we lost stuff. So stuff must have left the cell. Now, we've concluded at this point that the only thing that could account for that much stuff being added or lost is water. Cells are mostly water. When we see them shrivel up, it's because they've lost a lot of their water. They've gotten very dehydrated. When they start to puff up and even explode, it's because they've gotten too much water added. So we have to think about the fact that we just established that water doesn't cross cell membranes on its own. Water is polar. It's going to stay. If it's already outside the cell, it's going to stay there with the other polar things. The water inside the cell is going to be interested in the other water molecules inside the cell. And then that fatty, waxy inside of the cell membrane layer just isn't going to be interesting to water. So it's not going to go in there. It's just going to be indifferent to the inside of the cell membrane. And yet what we see when we stick red blood cells into different kinds of solutions is that water clearly is moving. It's either we've got water coming into the cell, flooding into the cell, and then we get the puffed up cell that could possibly explode. Or we get water leaving the cell, and then we get this shriveled up cell that could potentially die. So that means that actually the cell is not even controlling the flow of water. Because you can imagine that if you're a cell, it would make sense if you start to puff up so big that you might explode to shut down the water flow, to not let any more water in. And same thing, if you're starting to shrivel up like this and you're losing water, well, why don't you shut down the water escaping? Why don't you trap it? You're like, no, this isn't good. We're getting too shriveled up. We're going to die. We should stop letting the water out. So what that means is when we look at a red blood cell or any cell that we're talking about, what we observe when we watch what happens when we put the cell into the various different solutions is that there must be, so I'm just going to draw these little guys all over the place. If we're seeing that water is freely able to move, even to the point of destroying the cell, so the cell can't control water flow, so much so that if you put it in a certain solution, the cell will puff up and explode and be dead. In other cases, it can shrivel up and get so dehydrated that it's dead. So that must mean that the cell does not have closable gates for water. Otherwise, it would get some sort of signal that it's either getting too puffy or too shriveled, and it would shut down the water flow. So instead, we must have these channels, because we know water doesn't just cross a cell membrane on its own, because it's polar, it's not attracted to the inside of the membrane. So we've got channels where water can come and go as it pleases. These are just open doors to water. And the interesting thing is, when you look at one of these channels, they're kind of funky shaped. So we talked about how they're made out of protein. And what you'll see is that there's some little positive charges along the inside of that channel. And that's the really cool thing, is that, so we looked at water and we're like, well, water's not going to be interested in being in the inside of the cell membrane. Like, so here's our cell membrane with a little phospholipid bilayer tails on it. We got these guys. Water's not going to be drawn to the inside of that cell membrane. There's nothing to pull it there. And that means that since the cell membrane is there, very often the waters can't feel any of this attraction to each other, right? So we're looking for some kind of an attraction that would draw them towards each other. But they can't feel that as long as there's too much distance between them, as long as the cell membrane is keeping them far apart. But if we look at our water molecules acting as little mini magnets, we've got this kind of, more negative end of the water molecules here, the oxygen end. And then we've got these positive charges that are showing up on this protein, this water channel that we've got here. So what we're going to see is now this water molecule not necessarily is so much attracted to the water molecules on the inside of the cell, but it is going to feel an attraction to this couple of positive charges there. And then by the time it gets halfway through that channel, now it's starting to sense this guy out here. So they're going to start drawing towards each other. So that's one way to overcome the fact that waters can't really sense each other across the membrane because of the distance between them. And also the fact that water is not drawn to the inside of the membrane because it's nonpolar, so there's nothing magnetic there for them to be pulled towards. So these channels that are built all through the cell membrane, they're all over the place. You'll find them everywhere. These are called the aquaporins. So a pore means like a hole or a channel through. So that's the pore part, and then the aqua means water. So these are water channels. They don't open and close. They're just open all the time. And water is free to come in through them or to leave through them. Whatever forces are pushing and pulling on the water, they can always go through those channels. So that's a good thing in the sense that the cell often needs water to move in and out. But if you put them into certain solutions, it can be fatal because the cell has no control over where the water is going. So you have to then be able to predict, all right, so if water can either come in, it can flood in through this channel, or it can leave through the channel, well, then the next question is, which way is it going to go in a particular situation? Is water going to tend to come into the cell, or is it going to tend to leave the cell? And that that part of making that prediction of, okay, is this cell going to tend to shrivel up? Is water going to tend to leave? Or is water going to tend to come in? That's the next question that you have to be able to answer.